Hello, thank you for the introduction, and welcome to my presentation on protein DPI production. I am going to focus on the different destabilizing influences during spray drying. In the last years, protein drugs became more and more important. Those drugs were usually solutions for injection. A more stable and non-invasive alternative would be a dry powder formulation for inhalation. Proteins in general tend to denature or aggregate. During the DPI production, especially the drying part can be challenging, and therefore we need to find stabilizing excipients for protein stabilization. In my study, I focused on the impact the different stages of spray drying have on my model protein chymotrypsin. I used the nanospray dryer and compared 3 different stages. First, I took samples right after pumping through the tubing system in a circle. There, the protein is exposed to adsorption stress to the tubing material, to shear forces, and shortly while pumping through the spray head to thermal stress. Next, I took samples after atomization prior to spray drying. It's a little too fast, sorry. Those atomized samples were additionally exposed to ultrasonic sound because the nanospray dryer used a membrane nebulizer, and they were exposed to shear forces and air liquid interface stress. And last, I took fully spray dried samples. Those were additionally exposed to thermal and dehydration stress. As stabilizing excipient, I investigated mannitol. Sugars and sugar alcohols in general are known to be good protein stabilizers, especially against heat and dehydration stress. To investigate this for my model protein, I conducted a stability study at 50 degrees. Here in red, you can see my results for the pure chymotrypsin samples, and in blue for the samples containing additional mannitol. We can see that the activity is still higher after storage, and therefore can see that mannitol does stabilize chymotrypsin. For the samples treated with the spray dryer, I first measured the enzyme activity. There I was only able to find a small decrease after spray drying and no real change for the other samples. In the more sensitive fluorescence assay, I was able to see more results. Here in purple, we can see the fluorescent dye ANS in water, which does not fluorescent. In yellow, we see it after we added the native protein. There we have an emission spectra, and in gray we can see the denaturated protein, and there the emission increases, and we have a blue shift of the maximum from around 490 nanometers to 475. For my samples, here in green, for the pumped samples, no change appeared in the protein structure. For the atomized samples in red, I was able to detect a small change in protein structure, which we can see from the small increase here. And this was even bigger for the spray-dried samples. And here we can even see a small difference between the lighter blue line with pure chymotrypsin and the darker blue line with additional mannitol. This has a smaller increase in fluorescent emission, which does show that the mannitol does stabilize the protein a little. In my study, I found that the different stages of spray drying have a different impact on the model protein chymotrypsin. For the pumped samples, I was not able to detect a change in structure. After atomization, a small change in structure appeared. Here, the mannitol did not have an influence. And for the spray-dried sample, an even bigger change in protein structure appeared. And here we were also able to detect a small decrease in enzyme activity and also a small stabilizing effect of the mannitol against the thermal and dehydration stress. I'm going to investigate this effect in further studies, and I'm also going to investigate other stabilizing excipients against the other stress factors. Thank you for your attention. I'm happy to answer your questions.
Thank you, Friederike. Very interesting talk. I guess the first question is, I've had a few questions come in on the chat. One of the questions that we have had is, any idea of what excipients could be used to maximize the stabilization of the proteins that you're looking to spray dry to maybe also support scale up?
Well, it's also known to use amino acids, like I already tried glycine, but that's not finished now, so nothing more to tell. And we can also use polysorbates, but that's not so easy to spray dry. So there are a lot of other excipients in the line, but I did not try most of them. You can also use other sugars or sugar alcohols. But they all have different problems.
Thank you for that. Just one question, just on your sort of methodology. This is based solely on nanospray drying. Is that correct? Did you look at sort of more conventional spray drying?
I did now. I tried similar experiments with a mini spray dryer, which I think it's more conventional than the nano spray dryer. And there I found similar results, but they are not finished now. So I cannot tell for really sure.
Fantastic, thank you. Is that something you expect to see a difference in between the two different techniques?
Yeah, I think, I'm not sure if we can see a difference for chymotrypsin, but I think we can see a difference for different proteins, because we have other stress factors, we have other intensities of stress factors. For the nano spray dryer, for example, we have the ultrasonic sound, which is not that common for the mini spray dryer. And we have different intensities of shear forces and different intensities of air-liquid interface stress. For the nano spray dryer, we have smaller droplets compared to the mini spray dryer, for example. And for smaller droplets, we of course have more air-liquid interface stress. And so I expect to see differences, but I'm not sure if we can see differences for every model protein.
Fantastic, thank you. You have a question, Michelle?
Yes, a couple of questions. There's actually a few more questions come through. So one of them was about, can you tell us a little bit more about the stability that you did? You obviously showed some data at 50 degrees. Was there a particular reason for looking at 50 degrees? Did you look at any other conditions? How did you do any further assessment of the material on stability storage?
Degrees work best. That's why I chose it. I also tried 40 and 45, but that takes way longer. And of course it takes way longer. And the effects were also there, but of course, when the protein is more stressed, I can see more stabilizing effects. That's why I chose a higher temperature. And I also tried 60, but there the protein was kind of dead after one day, so it was not so easy to conduct a stability study there. So I just chose what worked best for chymotrypsin. I think it's different for every protein you have to try out.
I'm just thinking to the enzyme activity itself. You've shown some interesting data there, but did you look specifically at enzyme activity as a result, and to see whether you denatured them to a point where they were no longer active?
Do you mean that? Or I'm not really sure what you mean.
I mean, you showed on one of your slides how different it was, but I wasn't clear. Was that the activity of the material?
enzyme activity. But therefore, of course, the protein needs to change a lot, or in a certain space or so, whatever interacts with the enzyme activity. And in the fluorescence assay, we were also able to see changes that did not really interfere with the protein activity. So there were probably smaller changes that did not interfere with the enzyme activity.
Okay, thank you. And I think one final question before we close. So I think what you're showing is that you're starting to sort of really look at this as a potential technology. Have you thought about how you could deliver these, in a formulation? What type of devices would you think would be necessary? And how could you moderate any potential shear effects of aerosolization or through that delivery process on actually the functionality of the proteins that you're formulating?
Well, I did not look into that now.
Quite early.
So I mainly look into the production side. Yeah, probably the protein will also be influenced by shear forces during the inhalation. I think that mainly depends on the protein. There you probably need a very unstable protein, which would probably not be a good model protein. So it would probably be a good idea for another study or for another theme.
Error, I suspect. Lots of opportunity for further research.
Definitely. Well, that brings us nearly to the end of time, I think, Friederike. So thank you very much for giving this flash presentation. And we hope to see how your work develops over the coming years.